We discussed about different kind of omics data, and we will continue with uh, the methods, some of which we discussed yesterday, including BLAST, that are used for analysis of this data. But we are just going to focus on the methods that were developed for comparisons of DNA and protein sequences. Okay. By 1950s, methods or technologies were developed for sequencing proteins. Have you heard about a famous scientist named Frederick Sanger? You have? So what he did? Sanger sequencing, yes. And he got 2 Nobel Prizes, both in chemistry. The first one was for sequencing protein, and the second one was for sequencing DNA. Okay. Around I think 1953 or so, he developed the technology for sequencing proteins. And do you know the first protein that was sequenced? Which protein was it? Insulin, very good, insulin. So insulin was sequenced, and because of that, actually, it was a big thing that happened around that time, 1950s. He got Nobel Prize in chemistry, I think, as I remember, 1958. And by that time, 1950s and 1960s, many proteins were sequenced. And when I say many, still I would say in dozens or maybe hundreds, not like today where you have access to millions to billions of proteins. So I would say there were a handful of proteins that were sequenced, and people were very curious to compare them because they were coming from different organisms. So they wanted to compare these sequences to understand how similar or dissimilar they are, and how relationships among organisms can be inferred based on this comparison. Okay. And by that time, algorithms were not developed for aligning, because given 2 sequences, you would want to assess how similar or dissimilar they are. And one of the most used approaches is alignment based. Of course, there are approaches that can compare sequences without performing alignment, but alignment is the most used technique or approach for comparing sequences. So alignment methods were not developed around that time, and scientists were just manually aligning the sequences and assessing how similar and dissimilar the sequences were. So where did I keep it? Okay, so pairwise alignment in the 1950s. So you can see the alignment of 2 proteins, and these are hormones, beta corticotropin from sheep and corticotropin A from pig. And given sequences and being asked a question how similar or dissimilar they are, you would want to align so that you maximize the identities. Identities are positions where identical residues are aligned. So you want to align in a way so that there are maximum number of positions where identical residues are aligned. So basically maximizing the identities. So you can see identities here, glycine in second position, glycine aligned to glycine, and then there is one aspartic acid aligned to aspartic acid, and then glutamic acid aligned to glutamic acid, and then there are some mismatches. So the goal was to line up 2 sequences in a way so that the identity between them is maximized. Okay. And then another example. So at that time, sequences were being aligned by scientists more and less manually because programs were not developed for the alignment. So one can do if sequences are highly similar, but if they are dissimilar, highly dissimilar, so highly divergent sequences, it would be challenged to align them in a right way. Okay. So, but this was really interesting because by aligning these sequences, researchers were able to infer how similar, how dissimilar these sequences were, and based on that, how organisms were related. So, this is a very interesting work that was going on in the field of biology and molecular biology and of course evolutionary biology. Okay. So, pairwise alignment is the fundamental operation of bioinformatics, one of the most used operations in bioinformatics. It is used to understand how sequences are related functionally or structurally. So, how similar they are? If they are similar, then it is likely that their functions would be similar or related. And further, if they are related, then it helps in understanding how these sequences have evolved from a common ancestor. Pairwise alignment is used to identify regions in proteins that are conserved. Those regions are important because they are responsible for conferring functions to the organisms. So, these regions are called domains. So, you would want to use pairwise alignment to identify domains in proteins. Of course, we discussed about BLAST, so we will come back to it again. It is the basis of BLAST searching. And in 1980s and 1990s, the entirety of DNA in the cell of an organism could be sequenced. And the entirety of DNA is called genome. Genomes were sequenced in 1980s and 1990s, and still now it is one of the most fascinating projects because DNA is code, it's the secret of life. By sequencing, we are trying to understand about the organisms and also how the organisms are related to each other. Okay, so protein sequences could be more informative than DNA sequences when you want to perform alignment. So, can you think of reasons why you would want to consider protein sequences for alignment, and also some regions why you would want to consider DNA sequences for alignment? So, you have the options, you are given both DNA as well as protein. So, why you would want to consider DNA, and give some examples, and when you would want to consider proteins for alignment? Gene editing. So, how alignment will help?
For example, if I'm doing a gene knockout
Yeah.
some other organism, I would compare the sequence of human gene with the gene of that organism to see if it is compatible or not.
Okay, so one example. So, this is a DNA alignment. So, think about other examples. Why you would want to perform protein alignment, and why sometime you would want to perform DNA alignment. Okay, so we were discussing about redundancy in, you want to add something? Yes. SNP, yeah, SNPs. So, if you want to, which we were doing yesterday. So, if you want to identify SNPs, you would want to perform DNA alignment. And this is interesting because, for example, we would like to compare genomes of people here in this room. So, you would want to sequence and perform DNA alignment because it is possible that many of the proteins or almost all of the proteins could be 100% identical if our proteins are compared. Of course, there could be some differences, but most of the proteins could be identical. So where does the difference lie? In DNA, right? So, and if you remember that changes can happen in the third codon position without, most of the time, without changing the amino acid, right? So, an amino acid is coded by one or more codons. So, in some cases, if there could be multiple codons coding for an amino acid, changes in particularly in the third codon position will not result in a different amino acid. So, which means that DNA could change more, yet protein may not, right? Because changes in the third codon position may not result in a different amino acid. So, it means that proteins are evolving slowly than protein sequences. Protein sequences evolve slowly than DNA sequences. Okay? And of course, now we know that protein structures evolve slowly than protein sequences. We will see an example in a moment. Okay? So, so yeah, we are discussing about the choice that you may have and why you would want to perform protein alignment and DNA alignment, and you gave some examples, but I am still waiting to hear some examples of protein alignment. So, I will go ahead and start discussing that. So, how many amino acids are there? 20 standard, 20 amino acids. And so, you can think of protein as a polymer derived from an alphabet of 20 letters. Okay? While DNA is a polymer derived from an alphabet of 4 letters. So, you will say that there is more complexity with protein, more information with protein because of that. And also these amino acids have biophysical or biochemical properties, and they could be shared among these amino acids. So, they could have similar or related physicochemical properties. Okay? So, that is interesting because when you are performing protein alignment, then this information can be leveraged in the protein alignment, because if there are amino acids that have similar properties, in the course of evolution they may get replaced by each other without changing the function of the protein. Okay? So, that information is there. When you are performing protein alignment, then this can be then utilized. So, in continuation of the discussion that we are having, that proteins evolve slower than DNA, which means that if you have to compare sequences coming from very distantly related organisms, you would want to use proteins because proteins are evolving slowly. If you use DNA, because DNA is evolving fast, there could be saturation because so many changes will happen that those signals that can be used for inferring relationships may be gone for organisms that are distantly related. Okay? So, those sequences would appear more and less like random sequences because so many changes could have happened over the course of millions to billions of years of evolution. On the other hand, because proteins evolve slowly, it is possible that those signals would still be there. So, more conservation at the protein level, and so because of that there would be a longer look back time. Okay? So, that will help in inferring relationships among organisms that are distantly related. Okay, so that is important, and I think you gave several examples, and we discussed this, that an amino acid is coded by one or more codons. So, and because of this reason, there could be changes in the third position. You can see here. So, DNA can change, but protein won't actually, because these all are coding for glycine. Okay? That is why DNA could evolve faster while protein would not. Okay, we discussed all this, and you gave several examples. So why you would want to perform DNA alignment, and we discussed this, that if you want to compare genomes of or DNA of closely related organisms, then DNA alignment could be helpful. For example, if you want to compare the genome of modern human with the genome of ancient human, Neanderthal, then you would want to perform DNA alignment, because if you perform protein alignment, proteins could be 100% identical. So based on that, you cannot say anything about why they are modern and ancient, right? So that difference is not there at the level of protein. But that difference could be reflected at the level of DNA. So if you compare DNA, then you can see where the changes happen. Okay, single nucleotide polymorphisms, we discussed this. But if you want to compare regulatory sequences, non-coding sequences, they do not code for proteins. Genes that do not code for proteins, RNA genes, okay? So non-coding RNA genes. So for that, you would have to perform DNA alignment. And we discussed yesterday about cDNA and EST. EST is arising from a gene, right? So you have EST, you have sequenced. If you map the EST sequence to the genome, it would map to the source, right, from which it originated, and that source is a gene. So by performing alignment of EST sequences to the genome, you would be able to identify genes, okay, where the genes are, because ESTs are coming from genes. Okay, so these are reasons why you would want to perform DNA alignment. So pairwise alignment, this is the definition. So in the pairwise alignment, given two sequences, you line up sequences. So two sequences line up, all possible lineups of two sequences. You would want to select the lineup that gives you the maximum level of identity or similarity between them. This is the goal. Okay, you want to maximize the identity or similarity between two sequences. So just line up these two given sequences and identify the lineup that has the maximum level of identity or similarity between them. Okay, and of course, a similarity score would be computed. And if the score is high enough, then you may infer that the sequences are related. Okay, but then you would also want to ask what is the statistical significance of the similarity score. So methods have been developed to assess the significance of the similarity score. So that should be large enough to be significant, which means that that alignment may not happen just by random chance. That happened because they were related. Okay. Okay, homology. So you might have heard about homology, homologous sequences. So this is similarity that can be attributed because of descent from a common ancestor. So two sequences that are similar because they have descended from a common ancestor. Okay, so two sequences or organisms that have descended from a common ancestor, they are homologous to each other, and they are called homologs. Okay. So here you can see the beta globin and myoglobin homologs. They are these are the cartoons of their structures. So if you superimpose them, they superimpose very well. At the sequence level, they are highly divergent, but at a structure level, they are really well conserved. Okay, orthologs. So orthologs are homologs that has arisen because of speciation. Okay, so homologs are sequences or organisms that are related by common ancestry. Okay, so this is the definition of homologs. Orthologs are homologs that has arisen because of speciation. So new species evolve, and because of this, there are sequences in the different species, including the new species, and those sequences are orthologs. Okay. On the other hand, within a genome, for example, within our genome, a gene can get duplicated. Okay. So, and the duplicated gene and the original gene, they are of course evolving over a course of time. They both share a common ancestor, right? From where the other gene evolved, the originated and evolved. So, these genes that are related because of duplication event. So, homologs that have originated because of duplication, they are called paralogs. Okay. So, we will see, actually we will see some examples. So, here are examples of first actually is the example of orthologs. So, globin genes in different species, okay, and they originated and then diverged in different species because of speciation. So, they are orthologs, globin orthologs in different species. And then paralogs, so globins in the human genome, okay. Alpha globin, beta globin, cytoglobin, myoglobin, and neuroglobin in the human genome originated due to duplication, okay, and then they diverged and have acquired different functions or related functions. They are related to each other because of duplication, okay. So, they are called paralogs. So, you can visualize orthologs and paralogs in a single phylogenetic tree. And the phylogenetic tree can help you in inferring whether the sequences are orthologs or paralogs. So, let us try. So, there was an early globin gene in the last common ancestor, say, okay. And then a gene duplication event happened in the genome of that ancestor, resulting in alpha chain gene and beta chain gene in that genome, in the ancestral genome, okay. So, gene duplication, and then around this point of time speciation happened, and 3 species originated: frog, chicken, and mouse. And because of that, there is a frog alpha chain gene, chicken alpha chain gene, and mouse alpha chain gene, okay. And this is because of a speciation event that happened around this time. Similarly, mouse beta chain gene, chicken beta chain gene, and frog beta chain gene originated because of a speciation around this time, okay. So, if I ask you that mouse alpha and mouse beta chain genes are orthologs or paralogs, what you would say? Paralogs. And to infer that, you would want to trace back, and where they meet, that will help in inferring whether they are orthologs or paralogs, okay. So, let us actually trace back. So, mouse alpha chain gene and mouse beta chain gene, and I am tracing back. So, I want to trace back and see where they meet. So, tracing back, I am here, tracing back, I am here, they have not met yet, and then further tracing back and they meet here, right, and here was gene duplication. So, because of that, they are paralogs, okay. So, phylogenetic tree will help you in inferring whether 2 sequences are orthologs or paralogs. You just have to trace back and see where they meet. So, for example, if I ask you that frog alpha chain gene, chicken alpha chain gene, let us consider these 2 only, whether they are paralogs or orthologs. So, you will start tracing back. So, frog alpha chain, chicken alpha chain, okay, I am here and then I am here and then further here, right. where they meet. So frog alpha, chicken alpha. So tracing back and see where they meet. So chicken alpha will come here and then tracing back. So they are meeting here, right? And this is a point of duplication or speciation. Speciation, right? Speciation. So you will say that they are orthologs. So very simple way actually. First you have to make a tree, and there are methods to make a tree, and then trace back and see where they meet. And if the tree is annotated with duplication and speciation, then you would be able to infer which genes are orthologs and which are paralogs. Okay. Okay. Very good. Yes. Frog alpha and chicken beta. So trace frog alpha and chicken beta. So trace it, chicken here, you come here, right? And then here, where they meet. So what would be then? What you would infer? Paralogs, right? Paralogs. And I would not venture further into this. There are further classifications in paralogs and out paralogs. So and people sometimes they say actually, and many biologists also, that if genes are present in different organisms, they are orthologs. It is not so actually. You have to trace back and understand how they are related. What is the event that resulted in the evolution of these, and because of that they are related to each other. So that could be speciation or duplication here. Okay, clear? Okay. Okay, it could be confusing, and that is why phylogenetic tree will help you in inferring whether two sequences, whether even if they are in different organisms, they are paralogs or orthologs. Okay, so the general approach to pairwise alignment, you would want to pairwise alignment means two sequences. So you would want to select two sequences, and then an algorithm or a method that can help in aligning two sequences and scoring their similarity. Okay, so these are the steps. You would want to allow insertions because in the course of evolution, insertions or deletions can happen. These are represented by gaps. We will see in a moment. So you would want to insert gaps in the alignment, and those gaps represent insertions or deletions. A score, of course, is similarity. A score, it reflects the level of similarity between sequences. Alignments can be global or local. So global alignment means that the entirety of two sequences have to be aligned. On the other hand, local alignment means that only regions in sequences have to be aligned. That's why local. Okay, and then you would want to assess whether the similarity score that has been computed is statistically significant or not. So let's look at this example, alignment of two DNA sequences, and you can see that there are matches. So matches means identical residues or nucleotides here aligned with each other. Okay. So for example, G aligned to G, that's an identity or a match. On the other hand, mismatches are those nucleotide alignments where the nucleotides are different. So identities are shown by vertical lines, and the mismatches actually they are so you can see the middle rows, only the identities are indicated, and they are indicated by the vertical lines. So by looking at the middle row, you can assess how similar or dissimilar sequences are. But importantly, you would want to score the alignment of two sequences. So, and that can be done by computing the similarity score for this alignment, and which is obtained by summing the score for the alignment of the residues at each position. So, the score for this is sum of the scores for identities. So, all positions actually. So, if you look at the first position, this is a gap, A aligned to a gap, okay. Second position, T aligned to a gap, and so on. So, you have gaps. Gaps are insertions or deletions that can happen in the course of evolution in sequences. And then there are mismatches. So, C aligned to G, mismatch, and there are matches or identities. So, there are scores associated with each of these: match, mismatch, and gap. So, you would want to add the scores for these, and that would be the score for the alignment. And I am not going to go into further detail of this. So, there is a mathematics behind this, why you have to sum this up. But this is how it is done. This has been derived using mathematical expressions. So, and I got the screenshot for you from BLAST. For pairwise alignment, often you would want to use BLAST. It is one of the most used algorithms for sequence comparison. So, select BLAST and then select protein BLAST, and we would want to align two proteins. One is beta globin and the other is myoglobin. So, you would select align two or more sequences, okay, and then you would input in these two boxes the accession numbers of these proteins. So, the first one is beta globin, the second one is myoglobin, and then BLAST. You would also want to check the algorithm parameters, and these are the default values. You may not want to change unless you have understanding of these parameters and the reason why you want to change it. In most of the cases, if you do not know, you can use the default setting. So, and then BLAST. So, when I did this, it said that no significant similarity found. For regions, click here, and the reason is that they are highly dissimilar sequences. Beta globin and myoglobin, I showed you the structures, they superimpose very well, but at the primary sequence level, they are highly dissimilar. So, what I did actually, I edited the search. So, I went back to the algorithm parameters and I increased the expect threshold value from the default, which was 0.05. So, this is like significance threshold. So, you might have done some work where you had to compute significance of some experiment. You have to have a significance level. Often people use 0.01 or 0.05. Okay. So, the default is 0.05, and what I did, I increased it. So, basically I am relaxing the stringency so that the BLAST returns the alignment. So, when I did this, it indeed returned alignment, and it shows that percent identity is only 16%. So, 84% are mismatches, only 16% are gaps, only 16% are identities. Okay. So, which is shown here, the alignment of these two sequences. In the middle row, you can see the identities, the matches. So, that are shown by the one letter code of amino acids. So, you can see D aligned to D, look at the middle row, D aligned to D, D, and then E aligned to E, E, and so on. Okay. So, percent identity only 16%, positives I will get to this in a moment, and then gaps. Okay. Okay. So, So if you have to compute the score for the alignment, you have to consider the score coming from each position in the alignment. So let's consider a cross section of this alignment of these two sequences. So just this region, and for each position you have to get the score. So this could be coming from some data, some information already provided to researchers. So B aligned to B gets some score, okay, and that is 4. T aligned to L gets a score of -1, and so on, okay. So scores for matches. Matches means identical residues aligned against each other. Mismatches, which means different residues aligned against each other, and then gaps. So so all these scores have to be added, and that would be the score for the alignment. Okay. So you can see actually some values that may surprise you. For example, L is aligned to B here and gets a score of 1. You would imagine that if there are mismatches, then that should get negative score. But here we are seeing that there is a positive score for L aligned to B. Can you think of a reason why this should have a positive score? I'm sorry? Same, right. So leucine and valine, they have similar properties, which means that in the course of evolution, they can be replaced without altering the protein. Protein function will not change, so they can get replaced. So this is happening more frequently than you expect by random chance, right? So because of that, it gets a positive score, and there could be many such mismatches that can get positive scores, and here you can see many mismatches that got positive scores. And so when you are computing, you have to add the scores for matches. For mismatches, mismatches can have both positive scores and negative scores, and the sum for gap penalties. So there are only two gaps. The first gap gets a negative score of -11. The second gap gets a negative score of -1. So can you think of a reason why gap start has a stronger negative score than gap extension? Mutation? Mhm. Uh, yeah, so yeah, a simple way to say is that when insertions or deletions happen, often there are insertions of multiple residues or deletions of multiple residues. So which means that occurrence of an insertion or deletion gets greater significance than extension. Because if it happens either insertion or deletion, multiple residues are inserted or multiple residues are deleted. So the first one is really important because of the evolutionary significance of the event happening in an organism, okay, and the next ones they follow. So because of the greater significance of the event happening, the first one gets stronger negative score and the next one gets less stronger negative score. Okay, clear? Okay, yes.
On what basis is the
Oh, okay. So yeah, there is a whole work on this actually. So theoretical work, what should be given to the gap creation and what should be given to gap extension. Um, if you are interested, you would want to take a look at uh, work that was done many years ago, and that is in the textbook Biological Sequence Analysis by Durbin et al., Durbin and co-authors. And sometimes it is just based on heuristic approaches. So trial and error and find the values that make the sense when you are doing all these experiments. Okay, so there may not be theoretical or mathematical underpinning to these values. It might have been set or established based on heuristics. When people are using different values, then which one makes the most sense, which is the best one. So often in omics analysis and bioinformatics, heuristics are used when a theoretical approach cannot be implemented. Okay. Yes. Oh, how this table is created? Oh, okay. So if you look at, so these values, right? So these values are coming from a matrix which is called a scoring matrix or substitution matrix. So V aligned to V gets a score of 4. It is coming from the matrix. So there are 20 amino acids. So you will have 20 by 20 matrix. Okay. And these values are there. I will show you the matrix. So V aligned to V gets a score of 4. So which is here. T aligned to L gets a score -1. A aligned to N gets a score -2. L aligned to B, and this is showing here positive, which means that it will get a positive score. Okay. So L aligned to B gets a score 1 and so on. So you will, so in this row you have the scores for matches, and matches means identical residues aligned to each other. So sum all these scores. So this is sum of matches. In this row, you have the mismatches, and mismatches are residues at different positions in the alignment. So residues that are different from each other, okay, aligned against each other, but they are different from each other. So they may get negative scores or they may get positive scores. Positive scores when they share similar properties. So add these up. This is the sum for mismatches. Okay, and then gaps. So gap extension gets a stronger negative score -11, and then there is a gap extension -1. Sum these up. This is the sum for gap penalties. Add them up. All these three. It is the sum for the alignment. Okay. Okay. So yeah, these scores are coming from a scoring matrix, and we discussed about identity, similarity. So similarity includes identity as well as the mismatches that get positive scores. And conservation is, of course, when residues are evolving and they are changing, yet the scores are positive because those residues share similar physicochemical properties. So that's conservation. Okay, so here is an example of pairwise alignment of retinol binding protein. Retinol, you know, what is that? Vitamin A. Vitamin A. So this protein binds to vitamin A. So retinol binding protein, and another one beta lactoglobulin. This is a milk protein. and these are lipocalins. So here is an example, and you can see that there are identities but not many, right? In the middle row, identities are shown by vertical lines, and conservation is shown by dots. Okay. So this is an example where these sequences seems to have evolved and have diverged from each other since evolving from a common ancestor. Okay, because you observe fewer identities that are indicated in the middle row. Okay, so there are internal gaps. So you can see gaps, for example, here, and then there are terminal gaps. Here is an alignment of retinol binding protein from human and that of rainbow trout. And you can see that there are many identities. So look at the middle row and fewer gaps. So this is an example of high conservation. So if you are comparing sequences, pairwise alignment of sequences, then you are comparing sequences that might have evolved a long time ago. For example, if you are performing pairwise alignment of plant and animal sequences, then you are looking back 1.5 billions of years ago when plants and animals shared a common ancestor. Okay? If you are comparing origin of life, if you are comparing bacterial and human sequences, so basically you are looking back billions of years, so 4 billions of years ago when bacteria and human shared a common ancestor. Okay. Okay, so and here is an example of extreme conservation, and this is the multiple alignment of glyceraldehyde 3-phosphate dehydrogenases. And you can see that this is quite a high conservation. There are fewer gaps and fewer mismatches. And these are coming from organisms that are as diverse as fly, human, plant, bacterium, yeast, archaean. So, and this is an evidence that this level of conservation cannot happen by chance. So, human and bacteria must have shared a common ancestor. Okay, and this came because of sequencing. When DNA and protein were sequenced and then compared using protein alignment, then we came to know that we have all evolved from a common ancestor. This just cannot happen by random chance. Okay. Okay. So, and we were discussing about where these values come from when we have to score an alignment. So, I said that these values come from a scoring matrix or substitution matrix. And one of the first substitution matrices, and perhaps the first, was PAM matrix. Okay, and this was the first model of protein evolution that was developed by Margaret Dehoff and colleagues in 1960s, and it was a development that drove the field of molecular biology in a direction that, as you know now, the field of omics actually that is impacting many different domains of life sciences and medicine. So this was a very important development, PAM modeling. And I will not go into again mathematical details, but different PAM matrices were derived and they were numbered. PAM1, for example, is a matrix that reflects 1% divergence, which means 99% of the sequences are similar or identical to each other. On the other hand, PAM50, say, PAM50 will reflect 50 changes 50 changes over 100 residues in the alignment, and so on. Okay. Okay. PAM 250. 250 changes over 100 residues in the alignment. Okay. Possible or not? 250 changes over 100 residues. No, possible. Okay, so okay, let's say DNA. A change to T, T change to A. Okay, so how many changes? 2, right? But when you will perform alignment, you will not count it because A aligned to A. So back mutation. Okay, back to the same. And also there could be multiple changes at a position, right? While there could be no changes at certain position, they are just conserved. So if you count all these changes over 100 residues aligned against each other, there could be 250 changes that could have happened over 100 residues, right? Because of back mutation and changes, multiple changes that happened at the same position, right? So these matrices are reflecting those changes that happen in the course of evolution. Okay, yes.
How will you identify whether these residues function or back mutations?
Oh, oh,
There is a residue A to A.
yeah.
How will you identify whether it is function or A to T and T to A?
Oh, how it is? Yeah, again, there are models actually, and that is why that's molecular phylogenetics. So models have been developed that can help in inferring the changes that cannot be observed. Okay, so these are something that may not be proved in laboratory because they already happened. Actually, sequences are given to you, you have to infer. So mathematical models have been developed that can help you in inferring the unobserved changes, that cannot be observed. When you align it, it will be A to A. So there are observed changes that you can observe, and there are unobserved. So mathematical models can help you in inferring those changes that cannot be observed in the alignment. Okay, and that is important because unless you do this, your inference may not be robust. Okay, so PAM matrices were derived from closely related proteins because at that time there were limited number of proteins, and the proteins that were closely related, they were considered, they were greater than 85% similar to each other. So identity was more than 85%, and if you have such a high similarity, it is easy to align them, right? Because they would just appear so similar. It can be, and by that time, I think methods were already developed. So what they did is to construct a phylogenetic tree, and then based on that infer which changes happen. So again, here is an example of 4 sequences, globin sequences: beta globin, delta globin, alpha globin, and myoglobin. Okay, and Margaret Dehoff and colleagues, they used a method based on parsimony to infer ancestral sequences. So ancestor of 1 and 2, number 5, ancestor of 1, 2, 3, number 6, and this is the last common ancestor of all these 4. Okay, so they could infer the ancestral sequences based on a method. Okay, so let's look at the alignment. So they perform multiple alignment. So 1, 2, 3, 4, these are the sequences of interest. So given sequences, and then the inferred sequences number 5, number 6, and the last common ancestor sequence is not shown. So let's look at the beta globin and delta globin sequence. And so beta globin and delta globin. So can you see a position that is a mismatch. So, okay, which position is mismatch? This one, right? S aligned to T. Can you see this? S aligned to T. So, you would wonder that what happened actually in the course of evolution. So, S aligned to T. So, whether there was actually T in beta that mutated to S, or whether there was S in delta that mutated to T, right? So, you would just wonder. And evolution didn't happen like that. Evolution happened from their common ancestor. Okay, so right, organisms are evolving from their common ancestor. So changes are happening with respect to the common ancestor. So let's look at the common ancestor. So the common ancestor of beta and delta is 5. So let's look at this position as T, and what is in the common ancestor? 5. It is T, right? T. So which means that in delta actually no change happened. T to T, right? So it's invariant. So the change happened in beta from T to S. Okay. So now we know what happened. So the mutation is T to S, not S to T. Okay. And this could be possible because of inference of the ancestral sequence number 5 here. Okay. So this is how what they did actually. Okay, we'll come to that in a moment. They counted these mutations, and these mutations were called PAMs. PAM stands for accepted point mutation. So it is APM, accepted point mutation, but they changed it to PAM because it is easy to say than APM. Okay, and these are accepted point mutations, which means that they got accepted over the course of evolution, because that's why you are observing these, right? So they were accepted over the course of evolution, and they looked for these PAMs in different protein families. So for example, immunoglobulin kappa chain has 37 PAMs. PAMs are mutations, accepted point mutations, over 100 million years. Okay. Kappa casein has accumulated 33 mutations over 100 million years. Highly diverse. So, okay, so I will show you actually. So if you, for example, compare the kappa casein that has accumulated 33 PAMs over 100 million years between human and mouse, the percent identity is only 33%. So 67% are mismatches or gaps in the alignment, right? So highly diverse. On the other hand, some protein families evolved very slowly. They still are evolving very, very slowly. For example, ubiquitin has almost 0 PAMs over 100 years. So if you compare ubiquitins from human and mouse, percent identity is 99%, so highly conserved. So this way they were able to quantify the PAMs in different protein families, and that helped in understanding which protein families are evolving fast and which are evolving slow. And there could be a reason why this is happening. Okay. Okay, so here is the score actually that we discussed, and you wanted to know how those values have been derived. So those values, score values for two aligned residues, they have got those scores from this formula. Okay. And this is the score for residue I aligned to residue J. Okay. And Qij is the probability that I is aligned to J because of them sharing a common ancestor because they are related. On the other hand, Pij is the probability that residues I and j are aligned against each other just by random chance. Okay, so that's why this is called odds ratio. So this is what is in parenthesis. This is called odds ratio. The probability of two residues aligned against each other due to relatedness relative to just by random chance. So that's odds ratio. Take the logarithm of this. This is log odds ratio, and this is the score. Okay. So the values were derived using this formula, and this is the same thing. So we'll not go into this. So what Dayhoff and colleagues did actually, and we discussed already this, that they inferred the common ancestors and then they recorded the mutations, the accepted point mutations. So and they are shown here. So for example, if you consider the E to D mutations, okay, and let's say that this is ancestral and this is descendant. So E to D. So how many E to D are there total? 2. So E to D, ancestral E to descendant D2. Okay. So they filled it up, and this is the count matrix, and then they use the counts to compute the probability of a descendant residue B given the ancestral residue A. So the probability of B arising from A. Okay. So A to B mutation, and this can be derived using the count matrix. Okay. And this is not difficult to understand. Intuitively, you would be able to understand this. So probability of B given A means given ancestral residue A, which means that count of events where ancestral residue A is being replaced by any residue given A. Okay, replaced by any residue. So out of all these events, how many events are A, ancestral A, replaced by B? Okay, and this quotient or this fraction is the probability of descendant residue B arising from ancestral residue A. Okay, so they computed this because it helped in extrapolating the matrix that they had derived. You remember they derived a matrix from highly similar sequences, more than 85% similar to each other. But they wanted to derive a matrix that also represent highly divergent sequences. And this mathematical property arising out of this conditional probability help them in deriving matrices that reflect highly divergent sequences. Okay, so PAM1 1% divergence, so highly similar, but using the mathematical property from a well-known theory in mathematics called Markov chain theory, they were able to extrapolate from PAM1 to higher PAM numbers. So PAM 250 means 250 changes over 100 residues. So this is the scoring matrix. So it is 20 by 20, but it is half matrix because a score for L aligned to B would be same as B aligned to L, right? So that's why half matrix. So here is the scoring matrix, and these values are derived using the approach that I just described. Okay, and they are representing the scores for alignment of two residues. Those could be matches, mismatches, and of course there are then scores separately for the gaps. They were derived separately. Okay. Okay, so if you can see, and this is PAM10. So what does PAM10 signify? 10 mismatches, yeah, 10 changes over 100 residues, and also this can be interpreted a bit differently. So PAM1 means one evolutionary time unit. So changes that happened over one evolutionary time unit, and that evolutionary time unit could be of the order of millions of years ago. I don't know what is that, but it may not be few years. It could be evolutionary time scale. It could be millions of years ago. So PAM10 also reflects 10 evolutionary time units. So as you increase the number, for example, PAM250 indicates 250 evolutionary time units, and that could be in billions of years. Okay, so sequences that are highly divergent because they shared a last common ancestor billions of years ago. So that is reflected in PAM250 or higher PAM numbers. So this is PAM10, and if you look for the score for tryptophan, which is W, with T is no, W is tryptophan, T is threonine, yes, right. So and that gets a score of -19. So if you look at the score for WT mismatch in PAM250, it gets a score of -5. So it's more strongly negative in PAM10, but not in PAM250. Compared to PAM250, this is more strongly negative, right? So why it is so? What PAM250 is representing? Yeah, 250 changes over 100 residues or 250 evolutionary time units, which indicate highly divergent sequences. So this matrix should be more forgiving to mismatches, right? Because it is representing highly divergent sequences, sequences that shared common ancestor billions of years ago. Okay, and that is why this has less strongly negative scores for WT mismatch, okay, compared with this one, which is more strongly negative, because this is PAM10 representing similar sequences. Okay, very good. Yes.
So in what case they will take PAM and what
That's a good question, and this is why these yeah, it's still going on.
So
So
I think we can take a pause.
okay.
और उसके बाद
Okay, then I can continue. Okay, so I think lunch time for everyone, and then we will come back and we will continue. Okay.
take a break. Okay, and we start around little early. Okay, 2:15 around.
Okay.
In case of 2:30, we will start 2:15, and we try to finish by 3:50. Okay, so that we get more 30 minutes for discussion. I will continue with discussion.
Okay.
And then you can go.
Okay. Yeah, it's
for
okay.
your other assignment.
Yeah, okay, that's fine. Yeah. So we'll finish and we'll also do some tutorial, and then then I can leave. Okay.
अभी तबीयत सबकी ठीक है she is here. She wants some help. Medical help. Or health center.